Now I'd like to introduce today's speaker, Dr. Toni Piaggio. Toni is a research molecular biologist at the USDA APHIS Wildlife Services at the National Wildlife Research Center in Fort Collins, Colorado. Her research studies focus on developing genetic approaches to inform wildlife damage management efforts. Population-level investigations can determine geographical boundaries of populations, gene flow between populations, population connectivity, and genetic diversity within populations. Phylogenetic studies can test hypotheses of taxonomic definitions and evolutionary relationships. Research gathered in any of these areas can enhance effectiveness of management efforts. Because wildlife genetics data can elucidate population dynamics in host populations, they also can be useful in the management of wildlife diseases. Toni, thanks a lot for your willingness to share your work with us today. We really appreciate it. And with that, I'm going to give you the floor.
Well, and Dave, I wish you could give my talk. Your voice is so lovely to listen to. I feel like a squeaker after that. But thank you, everybody, for being here. And today I'm going to talk about monitoring gene drive vertebrates in the environment. This is something I've been thinking a lot about for the past couple of years. I'm going to give you a little bit of background on who I am and where I come from, where I've been in the gene drive space, and kind of where I'm thinking that we should be exploring and ideas for exploring that area. So I work for the U.S. Department of Agriculture, the Animal Plant Health Inspection Service, and also Wildlife Services, which Wildlife Services has an operational branch and a research branch. And I belong to the research branch, the National Wildlife Research Center in Fort Collins, Colorado, which is the green dot. We also have a bunch of field stations around the country that specialize in different research topics. But overall, our mission is to apply scientific expertise to resolve human-wildlife conflicts while maintaining the quality of the environment shared with wildlife. So we develop methods and information to address human-wildlife conflicts related to the following. Obviously, agriculture, we belong to the USDA, human health and safety, property damage. Primarily, we work in the world of human-wildlife conflict. And that can have a broad umbrella and encompass wildlife diseases, as we're all very familiar with in the past couple of years. I work a lot in the world of invasive species, which is my entree into the gene drive world. I also work in threatened and endangered species. And certainly my world is driven by the One Health framework. So this is my lab in Fort Collins, Colorado. And my swim lane is to apply DNA technology to wildlife damage management and wildlife disease issues. Today, I'm also wearing another hat. I'm wearing a hat as a founding member and board of directors of GBIRd, or Genetic Biocontrol of Invasive Rodents. And this is a broad consortium made up of NGOs, academic institutions in the US, Australia, and New Zealand, and also federal agencies within those states as well. So again, kind of to cover the area we're going to cover, it's a bit winding, but it does all fit together, and hopefully I will help you see that connection by the end, if not throughout. So I'll go over my role in gene drive space. I'll talk about a tool for monitoring rare and cryptic invasive species in the environment, and how we develop such a tool, how we quantify the uncertainty around that tool, and actual field implementation of the tool. And I'll do a little bit of deep dive on that. And at first, it may not seem entirely relevant, but the point is that it takes a lot of work to do this. And just like gene drive, it takes a lot of work. There's lots of things to consider along the way. And I think it's important for us to recognize that as we consider each new step, there are these complications and these things we need to consider. So then also, how can we apply this tool to gene drive monitoring in an ecosystem? Okay, so I know you guys, I've gotten to watch the other talks, and Dr. Dean Pearson, also from the USDA, talked about unintended consequences and the use of different kinds of models to figure out what unintended consequences in an ecological framework might be of a gene drive organism being released into wild systems. And you also heard from Dr. Gerka Cook, also of the USDA, and I appreciate the highlight of our agency. We do a lot of wonderful work. And she talked about targeting surveillance with risk assessment, so and pathway analyses. And because we've been kind of in the modeling world, I thought I'd put this up. I'm sure all of you are familiar with Austin Burt's 2003 groundbreaking work on the safety kind of concept of safety behind selfish genes to change the trajectory of a species on the landscape. GBIRd was formed, it says 2021, that's when our partnership charter was put into place, but it was actually formed in 2015, and the NASEM Gene Drives on the Horizon was issued in 2016. And we very much take that as kind of a playbook about having very slow and steady studies that build upon one another in a safety context. Let me see, I'm getting a lot of sun. There we go. Okay, and so building upon one study upon the other in the context of safe ways to test gene drive organisms. And also this work that arose out of that, which talks about potentially stringent confinement strategies for gene drive research. And we've been working, we have large animal facilities, testing facilities at the National Wildlife Research Center in Fort Collins. And we've been, because GBIRd focuses on genetic biocontrol of rodents and specifically mice on islands, we've been testing our biosecurity at NWRC for the hope that we might someday have a gene drive mouse that we would be able to test and do multiple studies on how a gene drive passes through a population in a captive setting. And then more recently, this work, the core commitments for field trials of gene drive organisms. So again, I'm sure all of you are familiar with this, but I wanted to point to how much work we're investing into thinking about taking gene drive organism out of being built into the lab, into lab trials, into field trials. And I'd like to push us a little bit further to start thinking about vertebrate organisms being released into the field, and what would we do then, and what do we need to know before that point? And also in thinking about safety mechanisms, certainly mature regulatory processes are a mechanism for safety, and we're very aware of that and engaged in this process early and often. Okay, so kind of what is our concern in this space, and why are we working on this when so many people are working on target malaria and mosquitoes? Well, we have a lot of concerns within our consortium because it's Australia and New Zealand, NGOs like Island Conservation, the USDA, concerned about mice and how invasive they are and how destructive they are to seed stocks and also to fauna as well. And so mice have been transported to islands around the world by humans very easily, and they thrive very well, and they cause lots of damage to flora and fauna that are not evolved to deal with them. And so as a population geneticist, I came to this space with a certain set of goggles on. And my goggles are that you have a very diverse population on, say, a mainland. I don't know, oops, whoa, shouldn't have done that. Okay, here we go. Wow. Okay, a very diverse population. Let's say this is a mainland population, and a few of these animals get on a boat and they take a ride and they get off an island, but suddenly you've subsampled this genetic diversity, and now you have a much smaller amount of the genetic diversity, and it's skewed, it's biased, right? Because of the way that you sampled. And then over time, some of these genetic loci, these alleles, go to fixation. And pretty soon, everybody in the population has this small a, small a. My idea, along with my collaborators, is that you should see this pattern on islands of what this founder effect is. So this subsampling, this biased effect, and that there would be alleles that go to fixation because of genetic drift and a founder effect. And so I wondered if these might allow us to look for island-specific targets for gene drive, and ones that might be adjacent to a PAM site that you need for, say, CRISPR-Cas9 or any other CRISPR-Cas pairing. Okay, so one of the methods, besides this one, wanting to explore if this was a possibility or not, we recognize that working on islands is a really nice contained way to test things like gene drive, a biocontained. And we also recognize that it takes careful site selection of islands in order to have that biocontainment, because humans have moved mice on and off islands before and will probably do it again. So how do you control that yet still be able to have this contained ecological setting where you might be able to safely test a gene drive? So we thought about size and how isolated it is, traffic, human habitation, and it's a critical first step to biocontainment. And in a minute, I'll show you kind of some of our processes of site selection. But first, we wanted to explore this idea of being able to spatially limit a gene drive by building a gene drive that was tailored to a target population, say on an island, through these, what we were calling locally fixed alleles. So alleles that were fixed on that population, on that island and that island alone due to founder events and genetic drift. So our approach was we sampled invasive mouse island populations and their putative source population. We did pooled resequencing or pooled seq. So we pooled and did whole genome sequencing of everything in that pool. And then we identified locally fixed alleles that were adjacent. We targeted the CRISPR-Cas9 binding sites and conducted population genetic analyses. So to bounce back before I tell you what we found is our key criteria for selection of islands. And we wanted an island within a country that had a mature regulatory environment, meaning really strongly and well-developed, meaning challenging, meaning that it's going to take us years to get through. But the plus side to that is some reassurance of that what we're doing is safe and has lots of eyes on it, and it has buy-in. And so the island is within a country or an overseas territory where most musculus populations are present and non-native. And so, you know, most musculus populations are from Asian countries, so that would rule out Asian countries or ones that are proximate to that. So the island is biosecure based on two key criteria, either closed to the public or has only infrequent or controlled visitation. And it's remote enough to avoid unassisted immigration or emigration of mice. And no challenges exist to the treatment of traditional rodenticide-based methods. So when we want to get rid of mice on islands, we use toxicants. And there's damage to the ecosystem, there's off-target effects. These can't be used on islands where there are large populations of people and their companion or livestock animals. And so you'd want an island that you targeted that also did not have that. And you also would want it to be where most musculus is the only rodent species present. And so if you're testing gene drive, people might have concerns that that can be transferred to another species to eliminate that fear, that possibility. But presumably you would have tested that already through contained laboratory breeding experiments. Anyway, so it would also need to be reasonably economical and feasible. Oh, the other part about it being the only rodent species is if you had to go in and use rodenticide, if something went wrong with the gene drive and you just need to eliminate the population, you wouldn't want another rodent species there. So the island should also have a maximum of 2 probable sources of introduction from a mainland or other island source to facilitate potential source population genetic characterizations. So we want something that we're pretty sure will have its own genetic signature. It's had time to evolve that. And so the first thing we did to look at these locally fixed alleles is we did some modeling. And I think we're all pretty aware now how valuable different modeling approaches are in this space and are going to continue to be to inform our next steps forward. So in the top graph is the island, and on the x-axis in both graphs is time, and on the y-axis on the left side is the relative population size, and on the right side is the allele frequency. And so on the island, we have population is fixed for a particular allele. We introduce the drive frequency, and this is, hold on, I had to write down my numbers. I never remember these. It's above a threshold release for 1.65 to 1. Is how we modeled it. You release it and it starts to take hold. There's no resistance that occurs and the population crashes because you have targeted this allele that is fixed within the island population. On the mainland, you might have a mouse transferred from the island back to the mainland, a gene drive mouse, 1 mouse per month. And let's say that the mainland only has 5% resistance. So it has 95% has this same allele that was fixed on the island that we targeted. But what happens is the drive comes in, but that 5%, selection acts on that 5%, you get a resistant allele that then goes to fixation. The population suffers a little dip and then bounces back. So this is our sampling that we did for the pool sequencing, the paired islands and source populations. We focused on mainland source, but in this case, this was Oahu and Midway. And we know that Oahu is the source for Midway, which is a military base. And what did we find? So these are 2 graphs that are measures of heterozygosity or genetic diversity. And the islands are in white and the source population is in gray. And you can see that each time we found less genetic diversity on the island, which bears out the theory, population genetic theory of a founder population. And we also used what was called a fixation index, or FST, to look at right now the connectivity between that island population and its source. And what was interesting is for most pairs, we found this level of 0.17. Which means it sounds like a low number because it ranges from 0 to 1, but actually it's significant. And so there's not a lot of gene flow. There is periodic gene flow, but it's quite low, but not as low as this. This was quite surprising to us, this 0.365. And so we dug in deeper, and we know there's mus musculus subspecies. So we did whole genome sequencing for each of the subspecies. And we found in Western Australia that the island was completely mus musculus domesticus, but on the mainland, it was a hybrid genome, which was really interesting. It was castaneus as well. Midway and Oahu were also hybrid genomes, but they were equally so. And so it didn't show up in the FST measure. So this is what led to the high FST, is they actually don't have the same genomic makeup, the mice on the island relative to the mainland. And I took this little divergence to say, this is kind of one of my stump speeches about how we need to learn a lot about any population, island population, or otherwise in a population genetic basic context, ecological, molecular ecological context before we do anything. And it takes time and it takes effort and it takes money to do. So these are the results of our pool seek. These are the filters that we put each time. So we did how many SNPs did we have? These are the total number of differences we found between the island and the source population. And then we looked at how many were fixed within the population. These are quite a number of alleles in these island populations that are fixed, which is super exciting. And then you start to put more filters on where are the PAM sites. Are any of these adjacent to the PAM site? Well, yes, they are. Is anything in a female fertility gene, right? If we're looking at wanting to use a gene drive and make sure all the offspring are female or all the offspring are male, or we want to target and be sure that females are not fertile, what would that filter look like? And down here, these are the source population allele frequencies that we would be willing to tolerate. And again, this 0.95 is what we modeled, that only 5% of the population does not have that allele and thus would be resistant. And we kind of dialed it down because ideally that, I think, would make us all queasy to think really the source population had 95% of the population had this same allele we were targeting for gene drive. And so we dialed it down. And as you can see, we do lose any of those candidate alleles. But there's lots of interesting information here about possible targets and ways we can go about targeting those. Okay. So overall, we've developed a method for exploring the existence of locally fixed alleles in a target population. There's many locally fixed Cas9 target sites were observed on these islands, but less than 2% of these were in genes with female infertility. And we identified 40 genes that harbored locally fixed allele Cas9 targets. And further characterization highlighted 3 potential candidate genes with attractive properties for population suppression. So we did have quite a few takeaways from this study. Whoops, sorry. Oof. The characterization of population genetics of a target population is critical, and it's a knowledge gap that takes years to fill. There's actually not a lot we know about mouse breeding systems, and they seem very plastic and very unique to different island populations. So the lack of basic biology for our target systems can vary per ecosystem. It's a critical knowledge gap, again, that takes years to fill. I think you see my theme there. So I took this little winding path and haven't gotten to environmental DNA yet, but it was for a purpose. I wanted to point out that in a gene drive vertebrate species, what things have we been thinking about? We've been thinking about safety mechanisms in working with such an animal in laboratory development, in trials to see how well that gene drive moves through a captive population. We've been thinking about how to spatially limit. Certainly the method I just talked to you about is the method we looked at, but there's starting to be a lot of exploration out there about how to set up a genetic mechanism, a biological mechanism to have spatial limitation. We also talked about island selection criteria. And something else I want to point out is stakeholder engagement and regulatory engagement. These things are both super important to ever evaluating whether this is something that can be deployed into the environment and whether it's safe enough and whether it's socially acceptable. So what if we are actually successful? What if we develop a gene drive mouse in our case, and we get buy-in? What if the public, what if there's an island population, a nation that says, yes, we want to try this on this island and we support doing this, and we get the regulatory approval to do so? What then? Well, we need to understand population dynamics on the target island, and we need to monitor gene drive in the population. So again, we have all these capabilities to model and estimate how many individuals we would need to release on the island and how often. We might have safety mechanisms for limiting if one of those gene drive organisms got off the island. But how would we know what was actually going on in that population? Well, something I wanted to put forward was environmental DNA. Environmental DNA is targeting fragments of DNA suspended in air, water, soil. It is low quality and it's low quantity. It's cells that are being shed by organisms as they move through or interact with the environment. It's primarily been used on aquatic species, and we've been doing a lot of work in the past few years and moving it into the use for monitoring invasive species that are semi-aquatic or terrestrial. And so we've been looking at Burmese pythons, Nile monitors, and feral swine within the US. So again, what we're targeting is fragments or complete DNA cells suspended. In our case, we've primarily been working in water, moving into soils and air currently. So other studies that target, oh, I already said that. Our goals were to test our ability to detect species-specific eDNA using first captive animals. Again, we have these wonderful captive animal facilities. So we're really blessed with that. We can take a Burmese python, for example, and dip it in a big trash can full of water and then sample the water and see if our assay we've developed can detect that ENA from that python. Same thing with feral swine. We have the capability and have done it with them, which is what I'm going to talk about today. We also need to assess our detection limits of such DNA. As you might imagine, this DNA is not homogeneous throughout the environment. So how do you assess if there's just a little bit of it, if there's a single, say, individual feral swine running around, or a single gene drive individual running around, or a single not gene drive individual running around? How would you detect that when the DNA is not distributed evenly and also maybe very rare and degraded? We also had a goal to make the field collection easy, but I'm not going to talk about that today. Okay, so why is this something that's useful in the context that I work in, which is in a management context? And in our goal with feral swine within the USDA, within APHIS, is to reduce feral swine numbers within the continental United States. And so there's some states we'll never be able to do that, but there's some states where we can go in and hopefully remove all the feral swine populations. But we need to validate that we have in fact we have eliminated them. And what if in a new state where we didn't think they were, someone calls and said, hey, I think I saw a wild pig in my backyard. How can we confirm that? So it can also help us estimate differences in population size before and after control efforts. It can help us monitor areas thought to be free of these invasive species. It's really nice for using in remote areas, particularly. We've eliminated a cold chain. You don't have to have complex equipment with the aquatic organisms. People often use backpack filters and filter the water. All we're scooping, pouring buffer, and sending it back to the lab. Because so feral pigs for us have been ideal because even though they're terrestrial, they're tied to water for their biology. They need to wallow for some periods of the day. So we developed a protocol to maximize our eDNA capture from these turbid waters. You can see from these pictures that the waters that they use, they like to wallow around and they stir them up, and they're quite turbid. And that can be a real challenge for eDNA capture. We also want to determine our limits of detection with this developed assay, and we want to determine how long feral pig eDNA can persist in the water. We also want to know how quickly we can detect it once the pig has touched the water. And then finally, we want to move to a field application where we can estimate our probability of detection. This is really important, right? We have this assay, we've shown you that it can work, but how many samples do you need to take to get to a 95% probability? And so we use occupancy modeling to do that. And then we have a quick demonstration I wanted to tell you about that we did in New Mexico. And so again, I'm going to get in the weeds a little bit on this assay development, but I think it's going to have a lot of echoes to the sorts of complications, intricacies of developing, say, a gene drive organism. and the things, all the multitude of things and the layers that you need to think about. And it also reflects on the fact that these are things we don't want to be doing when we get to the point where we might be releasing a gene drive organism. We need to think about the energy, the money, the time it takes into developing monitoring tools ahead of the game. Okay, so how do we optimize eDNA capture? So we're working these weird systems. Nobody has done this in before, and we know that they're really difficult for eDNA capture because DNA adheres colloidally to soil and the soil suspended in turbid waters. So there's all these different methods that we can use. I was going to point, but I lost my little mouse. Okay, so for DNA capture and purification, first you can do multiple ways of DNA concentration. There's multiple approaches to DNA extraction. And then there's ways that we can remove inhibitors after all of those steps. And then we have multiple choices for amplifying that DNA. So I'm not going to go into too many details on that part, but it's just to tell you that each one of these steps involves a multitude of small studies to figure out within that particular system you're working in, which one of these options are the best and which combination of those options lead to a well-developed assay. Okay, so then we want to determine the detection assay limits. These are pigs we had in captivity, and we wanted to know how long, say, does that pig need to come in contact with that water before we can make a detection. So we had captive feral pigs. We put a virgin tub into the ground. We allowed multiple pigs to wallow and drink in the water. We also had a single pig in a separate pen. And then we sampled every 15 minutes for up to 2 hours. And we took 3 samples at each time point. These are our results with the number of DNA copies over on the y-axis, and on the x-axis is the time of accumulation. Remembering our first sampling period was that 15-minute point, and immediately we detected the pig DNA, and we continued to accumulate DNA through time. And this was with the single pig. And obviously the curve was the same for a multitude of pigs. And we wanted to know how long this pig eDNA can persist in water. So we took samples after a week from these same pools, and then we left the samples in the environment to be exposed to UV and temperature fluctuations. And we took a sample every 12 hours for 2 weeks. And again, 3 samples per time point. And we took the water from the pen with the multiple pigs and water from the pen with 1 pig. And these are our results. Again, DNA copies and hours to degradation on the x-axis. The light gray is the single pig, and we could see pretty quickly that the DNA degrades. And so within 300 hours, we begin to not have very many positives. The group continued on. We had to use modeling to figure out how far out it would go, and our estimate is 72 days. This is a really handy tool because now we can say, okay, you think you eliminated all the pigs. We would love to take eDNA samples and tell you if we detect any, but we're going to need to wait 72 days at least before we go in and sample. Otherwise, we might be detecting DNA left over from the pigs that were there prior. Okay, and these are some of the work that we produced out of all of that. And then I talked to you about imperfect detection. So every monitoring tool, cameras, tracking, traps, have error rates, and they all have problems. Some animals get camera shy, some animals get trap shy. And so we know that eDNA is imperfect. You can take samples from this pool where this pig is, and some of them will tell you it's negative. Again, DNA is not homogeneous throughout the environment. So we wanted to see what our imperfect detection was. So we did continuous camera detection so we could know when a pig had been there. This was in Texas where we worked on Camp Bullis, which is a base that's fenced in and it's silly with pigs. So we knew our detection rate should be very high. We sampled multiple water bodies and different water body types across a couple months. So this is kind of our multi-scale occupancy modeling and the things, again, that we have to think about, the things that affect. Where do you sample in that particular pool? And what if you're sampling a stream? Where do you sample there? And where do you sample in the water column? At the top, medium, bottom, midline or bottom? What about, does pH affect it? Does a month affect it? Does, again, the depth that you sample? Then you think about all the things that you've introduced in the lab and all those effects that might contribute to your probability of detection. So we've used an inhibitor removal. Inhibitor removal is great at removing inhibitors. You also lose some DNA. So was it worth doing the inhibitor removal, or did we do better at detecting before we did the inhibitor removal? And then also across replicates, what is the effect of replicates? Okay, so what did we find? We found that our detection varied by month. Here's our probability of DNA availability, or probability of detection. And we sampled in September and October. November became hunting season, so that wasn't wise. And then there's a pH effect. And so we did better in September. We did better when our pH was at 7 rather than 8. And a water body type certainly had influence as well. And we did best in small streams in the fall. So they started to pool, and we did a lot better in pools instead of running streams, which makes perfect sense. And we also worked on trying to figure out what would be the best sampling scheme given all of these different influences within our model. And what we found is, again, here's our detection probability, number of samples on the x-axis, and that even for, our worst places to sample and our worst pH, if we did 20 samples per site, our probability of detection would be up at 90%. But you know what? That's a lot of work in the lab. So 20 samples per site, how many sites are you going to do? You get it into the lab, you're going to do multiple extractions from each sample, and then you're going to do your PCR replicates. And so you get exponential number of samples to handle in the lab very quickly, and it becomes really expensive. And so we developed an adaptive approach to minimize the cost, but not compromise detection. So we take all these samples, but we do them piecemeal. We'll do 3 samples first and all the replicates, and we'll see kind of if we get a lot of positives, we know it's time to go out and send, the troops into the field. There's a hot spot over here of feral swine. You got to go check this out. If we get negatives, we continue on through that iterative process to make sure that we aren't missing a low level of detection. And we've deployed this. Since I did this slide, we're using this in lots of states across the US, but our first go was in New Mexico, which was ideal because there were limited water supplies and there were these small pools across a large geographic area. And this was a very hard region for, say, trackers to look for feral swine because it is very large geographical region chasing just a few pigs. And it's a pretty inaccessible area, just dirt road, so it takes a long time to get there. But we worked with our partners, the Forest Service, and they have to go out and check these livestock ponds. So it's quite easy to put bottles in their hands and say, grab us 20 samples when you're there. And we, we're really, once we started working on the data, it was very obvious which spots were hot. And we were able to say, go out here. And the Wildlife Service operations took a chance on our results and put the expense into putting helicopters into the air, which is one of the ways to detect pigs and flush them from where they're hiding. And each and every time they were able to go out and find even just a few number of pigs and eliminate them from the landscape. So there are places that it's really, really successful. I would say there's ecosystems where it's not very helpful. And I'll get to that in just a moment. But kind of what we're working on right now is transferring this from a qPCR into a LAMP assay, or loop-mediated and isothermal reaction, so that we can put it on a lateral flow stick, which is like a pregnancy test or a COVID test, a home-based COVID test. Anyway, these are becoming much more widely used. And so we are doing this so that we could put these in the hands of the people in the field. So you don't have to collect the water and send it to us and wait for us to process the sample, right? Because pigs move, mice move, mosquitoes move. And so a real-time answer is much more helpful. And kind of thinking about how this applies to gene drive, certainly, you know, it's interesting to me that there are no mus or ratus eDNA assays. And also you think about the biology of these organisms and where they might live. They might live on islands where there's not a lot of available open water. And mice in particular get their water from plants. They don't go to ponds. And so how do we deal with that? And so we're trying a number of things right now, like swabbing plants in order to pick up DNA, soils. And we are thinking about a multiplex assay that we're developing right now. You could also do this where you could develop the assay that targets that gene drive, right? And so you can actually have the ability to sample the environment and detect the gene drive construct in that mouse or associated with mouse DNA. There's also a lot of handheld devices that are coming out. I talked about the lateral flow, but there's different versions of that also allow you to have multiple targets. And again, we could use this with field devices for if we wanted the qPCR because we wanted to be able to do a quantitative assessment, or there's the whole genome handheld field devices that we can use to start to look at diversity within the ecosystem or multiple gene drive organisms on the landscape. So that's pretty much what I have. This is my lovely lab members that work really hard and make me look good with the work that they do. Some acknowledgements, and then I think I can.
Yeah, Toni, you can just leave your slides up, actually.
Oh, okay.
Yeah, and let me just say thanks a lot for a great presentation, really clear. And before we get into the questions, let me encourage those in the audience to feel free to chat in your questions, and we'll be happy to take them. But as we wait for people to generate some questions, Toni, I had some of my own, and sort of some technical questions that had to do with the loci that you're actually monitoring for in these assays. I would assume, but your remarks about detecting drive loci suggests that you might not have to rely totally on repeat DNA. And I just was wondering, what are the loci that you actually use for your studies? Are they repeat DNA? Was it like ribosomal loci or mitochondrial DNA or something?
That's a great question. To date, we've targeted mitochondrial DNA. We have played around with, it would be really cool to be able to get individual ID. And so I have a colleague who's looked at using repeat loci, and to some success. We're using metabarcoding right now. So we're doing the single species assays, but we're also using metabarcoding in environmental DNA, looking for some native fishes in Colorado, multiple species. But those are all based off of 12S genes. And then we also have used site B and control regions. So primarily those standard genes.
But are you fairly confident that you'd be able to design this so that you'd be able to detect a gene drive locus, which will be probably a single copy, or in homozygous condition, 2 copies in a genome?
Right.
Big challenge.
Yeah, it is a big challenge, and Do those gene particles need to be what type of DNA do you need to have, and can you target RNA? That's kind of the next frontier. People are looking at RNA, which is wonderful for pathogens, and targeting those in the environment. So there's a lot of unknown. And what's exciting about this idea is that the envelope is being pushed right now on a lot of how do we apply things like capturing RNA from the environment. I was just organized a government-wide eDNA working group that has
Good.
Canadian...
Yeah, I wanted you to say something about that.
Yeah.
Maybe you could share some of the latest.
Yeah, so there's a government-wide eDNA working group, and we now have like 400 members, and we just had a workshop, and there's people from all around the world, crazily, and it was just my friend and I. He's in the Army, and I'm in the USDA, and we started this, I don't know, like 5 years ago, and it has been amazing to see the growth and the talks that people are giving and the envelopes they are pushing. Being in the USDA, I can't push as hard and fast, right? Because we only have a limited amount of money. So I got to kind of follow on the sexy coattails of what my colleagues are doing, chasing RNA. But what we do is the real solid work of getting it to the field, right? And really testing how well it works in an applied context. And that's what's been really exciting to me and where it's going.
Mm-hmm. So, from a technical point of view, what would you say are have been some of the major technical advances where things have been pushed that are allowing, this, the expanded use of eDNA in various domains? So, where are the technical advances coming that allow the technique to be applied?
I think. So far, what I've seen that people have actually accomplished is applying metagenomics, metabarcoding. One thing
Can you explain a little bit about that, metagenomics and metabarcoding?
Absolutely. So metagenomics is taking an environmental sample and using whole genome sequencing to sequence all the genomes captured within that sample. Metabarcoding is a little bit technically easier. Okay. You're taking a single gene and amplifying that for every single thing that's in that sample. And we got to use it in a really exciting way. Again, we work in an applied world. And if there's a disease outbreak, like back when there was Zika, remember Zika? And how scary that seemed? We needed to put out, in Puerto Rico, we needed to put out insect traps and monitor. That was the first place that there was microencephaly within the United States. And so we wanted to see what mosquito species were there, what they were feeding on, and if they carried the pathogen. So you put out insect traps. Well, insect trap, you catch tens of thousands of insects that then somebody who has the expertise to morphologically identify these different species has to sit and sort through. Then you're going to take the blood meal from those, and you're going to try and get the host DNA, and you're going to try and capture RNA before it degrades too far and see if you can find the pathogen. Well, what if you just take that whole bulk insect trap, grind it up and use metagenomics, sequence everything in there. And so we did some mock communities, meaning we took a known number of different mosquito species. We had a single mosquito in there that was infected with Zika, and we had another single mosquito that had fed on a cow. And we mixed it all up and we used metagenomics, and we were able to detect the whole genomes of the Zika, of the host, and of the insect species. And so that's how much time does that save us in being able to figure out, and in real time when you have a disease outbreak, something like that is really important. So that we consider that eDNA. The nuances of what is eDNA is a whole other topic, but basically there's the broad umbrella of non-invasive DNA. You can get this without capturing the animal. Obviously, insects are different. You're capturing the whole animal, but you're not capturing that individual that they fed on, right? And so that's called iDNA or insect DNA. And you can look at ecosystem processes just by looking at insects that feed on blood. What have they fed on in your ecosystem, and how often, and what time of year? I don't know. I
So
went
just
all
to
over the
pick up
place
on
on
that,
that answer.
yes, but to pick up on the insect part and to sort of probe a little bit about to all the people that are working on eDNA that you mentioned in this group, can mosquitoes?
Yeah, I mean,
Are
that's what we were.
people using it?
Yeah, so then the concept of mosquitoes, that falls into that iDNA, and looking at, within let's say a system that's targeted for gene drive, you want to know, well, what the role of the mosquitoes are within that ecosystem. So conceivably, you could put insect traps out on transects across a year, and you could investigate what they're feeding on, what their roles are, right? We know males are pollinators, females are the blood feeders. And so what is their role within that particular target ecosystem? Not just what we've learned from someone doing a study somewhere across the globe.
Yeah, but they also have an aquatic, larval stage, and
Exactly.
and so taking advantage of that might be possible too. And that was what. So I think there's some analogies there in terms of your pig story and mosquitoes, because, and in some cases, in the case of say human malaria mosquitoes in Africa, they live in little puddles and pools, and which very much resemble the pig wallows that you were describing. And also
insects can have inhibitors in them. So when you grind up insects, that can inhibit your ability to amplify DNA. So overcoming those technical issues, overcoming the issues, excuse me, of wanting to capture the pathogens also that these insects transmit. So how do you preserve that RNA right away? And then how do you capture it and capture the DNA? And what is the best technological methods to understand what are in those samples?
Yep. I neglected to ask a couple questions here from the audience. Actually, Hector has a question. It says, could there be other genes besides female infertility? This goes back to your to the drive component of your talk. Could there be other genes besides female infertility that could be likely targets for gene drive? And would that increase your likelihood of finding a suitable gene target?
I hope so. We hope to go back into these data and do some more data mining. That was kind of our first go. And, I'm kind of assuming at this point that it might not be a Cas9 target. And so we would need to go look at other suitable Cases. And the nice thing about collecting these whole genome data from these islands and their putative sources is it is this amazing library that we can continue to go back and dip into and ask different questions once the technological advances arise and we start to see where the genome editing is going with mice. Is it going to be a gene drive? Is it going to be a T component or what?
This concept of private alleles, or to exploit private alleles, is, I haven't heard it discussed, and that doesn't mean much in the realm of mosquito gene drive. But maybe that's because there's more gene flow in some of these island populations. I don't know, but
there was a paper that came out.
What's driving loci in many cases happens to be either the locus itself has an impact on the fitness of the organism, and so it's key to target that particular locus, irrespective of whether it's private or not. That locus is what's really important.
It can also be a random event, simply from low genetic diversity. But there is a paper on locally fixed alleles in mosquitoes, and it came out right around the time as ours. And I'm sorry, I'm neglecting who the authors are, but there is a paper.
Okay, well,
looking at that, and they did find some locally
exposing my ignorance in
No,
these
no,
are the kind of
no.
things I should know.
Well, I'm exposing my own.
Hopefully the
'cause
boss is not listening.
No. I'm exposing my own because I'm not remembering the authors and
Okay, well, I
where they
can go
did this.
back and look for sure, but yeah, it's not typically not something that's widely discussed. I know it's much more widely discussed in your domain than it is in the mosquito domain. Your idea of using point of care type technology, your lateral flow things is really interesting, and again, I think there's so many similarities to the kinds of problems that you describe with feral pig monitoring and the kinds of things that I see would be analogous to, say early field trials. Well, like you're talking about, a trial on an island or someplace where you want to make sure that it's remained contained. So, for example, in your island experiment, I suppose once that gets started, you'll have to monitor the mainland in some way.
Absolutely.
Because you've identified where the potential sources are, and then you'll have to, and so you'll be faced with this reverse problem of a gene drive mouse getting off an island becomes essentially like an invasive species, and you're faced now with the same technical challenges of finding those low density, low frequency events within a fairly large environment.
Excellent point. And we have put our single species assay for the feral swine onto a lateral flow strip, and it works. We're going to try and deploy it in some field test to test its limits of detection and kind of its capabilities in the field. And that technology isn't really to a point where you can multiplex different species, but there are devices being developed out there that would have multiple lateral flows on them. And so, it's the continual pushing of, from the way I see it is the top, like what is the next thing? Can we find RNA in the environment? Well, obviously we can, but can we preserve it quickly enough? Can we find enough of it to start to look at pathogens in the environment or how proteins work in these organisms? Can we then on our end, we need to push that applied science. Well, what we need is we actually need a handheld device that someone can go and wipe on a plant or dip in some water and detect what they need to detect. And so I see it as working both ends of the spectrum, right? And what I do isn't always all that sexy working on feral pigs and wallow water, but it has a real purpose, and we really discover what we still need to poke on and what we still need to develop.
I was, I guess, again, I was really interested to see how long that eDNA lasted in the environment, particularly in the multiple pig case, and because there's so much out there that could degrade it. But I just wonder if the, in terms of origins of life, people have thought about, clays and certain kinds of, soils and things like that, providing good environments, that nucleic acids could associate with during early evolution. And I just wonder if some of that murkiness of the water is actually facilitating the longevity of the DNA by providing some of these sites where it can bind and becomes really inaccessible to
degradation,
to degrading enzymes.
Yeah, no, you're absolutely right. And that is where the world of ancient DNA comes in. So their favorite thing to do, right, is to take soil cores, because DNA adheres colloidally and gets preserved. And there's different types of soil that do that better than others. And so we're able to, it's just fascinating to think about our environment as this palette of DNA that is fleeting through dispersion and diffusion. And there's DNA that's locked up for tens of thousands of years waiting to tell us about how woolly mammoths walked across the earth, or what saber-toothed tigers, what their distribution was. And it's just completely fascinating since this world has opened up.
Well, I don't see a lot of questions coming in from our audience, Toni, so I think it's just me and you talking, but I think
It's always a
we can
pleasure.
do that. Yeah, we can do that on our own, and we can probably let the audience go unless they have questions, and I would encourage them to submit them as quickly as you can, because if you don't, we're gonna wrap things up here. So I do think that to me, I see so many similarities. Of course, most of the things that I think most about probably happens to be mosquitoes, and that's certainly my background. But seeing, you know, the problems that you're working on, from my perspective, I see so many analogies and similarities that particularly this eDNA component of it. So I'm really excited about, you know, the progress that's being made there, and I'm really excited about sort of delving into that more and seeing what's being done on the insect side, and whether that could be applied to large scale area-wide monitoring for, you know, a gene drive product in Africa or something like that would be really, really awesome. So
I think there's
anyway, so let me,
absolutely possibilities and potential there. I am absolutely confident that's where we can go with monitoring tools.